This interview is with Simon Leedham, Professor of Molecular and Population Genetics at the University of Oxford and an Honorary Consultant Gastroenterologist at Oxford University Hospitals. His work focuses on understanding gastrointestinal stem cell biology and the morphogen pathways that control them in carcinogenesis. He examines the clonality and genetic mutation burden surrounding predisposition to colorectal cancer. I hope you enjoyed this interview as much as I did. Thank you. Welcome, Simon. It's great to have you here today. Thank you so much for taking the time to be here. So we'll start things off with how you got to where you are today. So what drew you to specialise in gastrointestinal research, in particular as a branch of stem cell research?
Hi, Martha. Thanks very much for the invitation to chat today. I started my scientific journey as a medical doctor. I trained as a medical doctor and did about 7 or 8 years of being a junior doctor in hospitals, and soon realised that that was really tough and a difficult in terms of family life and ways of working. So I decided I would like to take some time out of that to do a period of intense research, which I then did with a particularly inspirational character called Professor Sir Nicholas Wright at what was then the London Research Institute in the middle of London. And I did my research on stem cell biology with him, and I absolutely loved it. I became fascinated by the topic. I got really enthusiastic about the entire process, the entire investigation. It was the kind of beginning of the sequencing era, or at least the accessibility of the sequencing era. So we had a lot of fun new tools to play with. And I became very, very excited and interested and passionate about the work that I was doing. And that changed my entire career, because from then on, I knew very much that I wanted to become a scientific researcher primarily and a clinician secondarily. And I therefore then angled the rest of my career development towards that goal. So it was a real sort of transformative moment doing a period of research for me. I thought I was going to be a clinician, and I've ended up being more of a scientist than a clinician.
Yeah, it's funny how you go into something thinking it'll just be a short period of time, and then actually it really changes your path, which is great, really.
It is, and we're very lucky, and in the science and medical fields, to have the ability to be able to find something that really grabs your interest and diverts you down a route that you perhaps didn't anticipate you would go.
Mm-hmm. So you specialize in stem cells, and stem cell maintenance and proliferation relies on many different signaling pathways, with the Wnt pathway perhaps being the most well-known of these. So for our listeners, just to start off, would it be okay for you to give an overview of how this pathway works and what it is?
Yeah, absolutely. So the Wnt pathway is a fundamental signaling pathway in the kind of standard canonical signaling pathway mechanism. So you have a receptor, you have a ligand, and you have a signal transduction mechanism within the cell. So when a Wnt ligand binds to a Wnt receptor, it triggers a whole series of processes which then they sort of de-repress the central mediator of this pathway, which is called beta-catenin. So beta-catenin is your central mediator of the pathway. In the normal situation, without ligand and receptor binding, the beta-catenin is disaggregated, it's broken down. So therefore, it doesn't move into the nucleus and transduce the signal. When you bind your ligand to your receptor, you break down the central complex called the destruction complex, which is the thing that's destroying the beta-catenin. So you're destroying the destroyer, which means you free your beta-catenin to move into the nucleus and set up and do the downstream activation of the pathway. And that destruction complex, which is itself destroyed, as it were, it contains a few very important proteins, one of which is called adenomatous polyposis coli, and that is really the central gene that is the principal mutation that occurs in colorectal cancer. So you have a process by which you acquire a mutation in the adenomatous polyposis coli gene, and that then activates your Wnt ligand at the level of the signal transduction pathway. So at that point, it becomes independent of ligand receptor binding. Now, the important thing about the Wnt pathway is that it's involved in stem cell stemness, it's involved in cell proliferation, all of those things that you want to be able to control if you want to avoid cancer. So very importantly in the gut, it's that activation that occurs pathologically in colorectal cancer, and it's that that drives the transformation. I hope that's clear.
Absolutely, clear as. So there's an idea called the just right hypothesis of this Wnt signaling, which determines the APC, as you mentioned there, the mutation spectrum of this throughout the intestinal tract. And this optimal sort of Goldilocks level of Wnt signaling differs in different areas of the intestine. So why do you think this is the case?
Yeah, it's a fantastic theory. This was developed by one of my mentors, a chap called Professor Ian Tomlinson. It relates to the idea that there is too much beta-catenin, too much Wnt, and the cell proliferates too much and becomes hyperactive and dies, apoptosis. Too little Wnt and the cell doesn't divide at all. So you have this kind of just right, or as you say, Goldilocks amount in the middle, in terms of when you have a mutation, you develop the just right amount of Wnt that allows you to proliferate out of control without apoptosing. So it is this sort of fine regulation of this Wnt pathway and the dysregulation that occurs with mutation. And the fascinating thing about this, quite difficult to explain, but if the APC protein, the APC that we talked about just a minute ago, is a very big protein. It's something like 2,000 amino acids. So you have mutation in colorectal cancer that occurs along the length of that protein. But the bit where the mutation clusters, called the so-called mutation cluster region, is the bit where it gives you the just right amount of Wnt. So that sort of level of Wnt activation is then perfect for tumorigenesis. And what's really fascinating in some work that we did a few years ago is that we looked at the level of Wnt activation that allows you to develop tumors across the gut. And the level of Wnt activation in the colon, for example, is very different to the small intestine. And therefore, we think that the amount of Wnt that these mutations give you in the colon is the right amount to initiate tumorigenesis. And that's why we think part of the reason why we think that the colon is the place that you develop a lot of colorectal cancer. Whereas, interestingly, the small intestine is a much rarer place to develop cancer. And we think part of that is the sort of dosage, if you like, of Wnt that is present homeostatically and then is provided by the optimal mutations in that particular protein, APC.
Yeah, I see. And it doesn't just directly affect tumorigenesis in terms of tumor growth, but also it has a wider effect on the tumor microenvironment as a whole. So it can promote things like M2, TAM phenotypes, or deplete things like gamma delta T cells and things like this. So it has a really broad reaching impact on the whole tumor. And then alongside the Wnt pathway, there's other pathways which interact and crosstalk with it, including things like the Notch pathway or hedgehog signaling. And now your research is looking into the bone morphogenetic protein, or BMP pathway, as we'll call it from now on. So how does this BMP pathway interact with the Wnt pathway, and how does it affect its activity?
Yeah, it's a fantastic question, Martha. So I mean, you touched there on a really important thing that we're really excited about and interested about in a minute, which is this idea of the tumor as an ecosystem. And therefore, the idea that you have your epithelium, which is next to a load of immune cells and next to a load of stromal cells, and those cells are all interacting. They're all cross-talking with each other. And these pathways that are central in giving the epithelial cell a fitness advantage, like the Wnt pathway, like the Notch pathway, like the BMP pathway to some extent, they don't act in isolation. So when you disrupt the Wnt pathway in an epithelial cell, fascinatingly, you're quite right. You're also disrupting the interaction of that cell with its microenvironment. So we're really fascinated by the idea that a successful initiating mutation, a mutation that is capable of starting cancer, doesn't just have an effect on the epithelial cell. It also has effect on the stromal cells, on the immune cells, on all sorts of other things. And I think that kind of pleiotropism, that multifunctionality of these signaling pathways is really fascinating. Not just about what it does to the epithelial cell, but how it makes cells talk to their microenvironment. So the BMP pathway is a equivalently fascinating pathway to the Wnt pathway. If you like, I kind of think of it as being the yin to the yang of Wnt. So if Wnt is involved in proliferation and stem cell stemness, BMP is involved in differentiation and apoptosis. So at different ends of the intestinal crypt, where you have your stem cells at the base, you've got a very high Wnt gradient, and you've got a very high BMP gradient at the top of the crypt, where the cells are apoptosing and differentiating. So therefore, you have this very carefully and finely controlled balance and homeostasis. You've got your Wnt at the bottom, you've got your BMP at the top, and if those two gradients are settled and stable, you have regular homeostasis with stem cell division followed by differentiation and apoptosis. If those pathways are deranged, for example, if you downregulate your BMP pathway or you upregulate your Wnt pathway, that is when you start to have problems. You start to have loss of control of cell division, you start to have problems with apoptosis differentiation, and that's where cancer initiates. So what's so fascinating about all of the intersection of all of these homeostatic pathways is that they've all evolved in this kind of cooperative, collaborative system to be able to maintain homeostasis and the proper control of cell division. When it goes wrong because mutation intervenes, it disrupts that balance, and that's where cancer initiation is a problem.
And I've read that the SMAD4 and P53 status of the tumour come into play with this as well. So no, if they're mutated or depleted or whatever. So it's quite individualized to that tumour itself. Is that true?
Yeah, absolutely. So SMAD4 is the signal transduction mechanism for the BMP pathway and the TGF beta pathway. So these are two very important signaling pathways that converge on this transcription, on this signaling transduction mechanism, SMAD4 in the cell. So SMAD4, unfortunately, is a target for mutation. It's on a gene that quite often is lost, or an area of the chromosome that is quite often lost as colorectal cancer develops. So there is selective depletion of SMAD4, which means that those two pathways, those two important homeostatic pathways, are then fundamentally disrupted. And again, it speaks to this idea that the mutations that initiate cancer are the ones that disrupt these very important homeostatic signaling pathways. P53, as you know, is often referred to as the guardian of the genome. It's very important in maintaining cell cell apoptosis. So in sending cells that are mutated off to apoptose and to protect us from mutation. When that is mutated, it's one of the most commonly mutated genes in cancer, because when that is mutated, you've lost the guardian of your genome, and then you are able to tolerate, your cells are able to tolerate increasing amounts of mutation. So it almost becomes a snowball effect. When you lose P53, you tolerate more mutation accumulates, and the process of cellular disruption goes on.
Yeah, absolutely. And as you were saying, this careful balance between the Wnt pathway and the BMP pathway is quite, I don't know, because there's such a high level of control there. There's almost like it's on this seesaw, and it can very heavily fall one way. So the BMP pathway constituents are expressed by mesenchymal tissue, but then these interact and influence the intestinal epithelium. Do you think the fact that this is paracrine rather than autocrine gives a higher level of control in this homeostatic, healthy state, but then also perhaps as a downfall when it comes to cancer, because there's another cell type which can be altered within the tumor microenvironment?
Yeah, fantastic. I mean, fantastic question again. So this speaks to this idea of the tumor as an ecosystem. So we've had a very epithelial centric view of cancer for a while, in that we've always thought that it's the epithelium that is the cell of origin, which it is, and that mutation is the sole what's the word? The sole thing that's driving. So epithelial cell mutation is the sole thing that's driving cancer. I think what you're describing there is this idea that actually that the homeostatic regulation is more complex than that. It's not a cell intrinsic thing alone. It's not an epithelial cell intrinsic thing. We don't have regulation written into our epithelial cells. The regulation of the epithelial cell comes from this kind of multifunctional levels of control at the level of the receptor, control at the level of the ligand expression, control at the level of the signal transduction, and control at the level of the antagonists. And that is all about epithelial mesenchymal crosstalk. So the fact that these two fascinatingly embryological distinct compartments, we have the endoderm-derived epithelium, we have the mesoderm-derived mesenchyme, but those two in those two compartments in the intestine crosstalk to each other with extraordinarily large amount of interdependence. And I think that's really fascinating because you're right, it does provide a very sophisticated level of control at multiple levels in homeostasis, but it also means there are multiple targets that can go wrong in cancer. So for example, one of the things we've been particularly interested in is the overexpression of a stromally derived antagonist of BMP. When you overexpress that antagonist, that can end up with reducing the amount of BMP regulation, which increases your chance of initiating tumors. And I think you're right. It's that kind of fascinating interdependence between these cell compartments that we're really interested in studying.
So you talked about this antagonist, and we're going to delve into that in a minute. But just before that, I'd just like to delve firstly into your methods of study. So you've developed a technique to examine the mutational burden of a single intestinal crypt, which is then used to analyze clonal ordering across this single lesion. So can you walk us through how this works and what the term clonal ordering really means?
Yeah, so these are the techniques I developed during my PhD. It was what I loved so much when I was doing my PhD with Nick Wright was because I was taking a very epithelial centric view of cancer then, as really was the sort of trend of the day. I mean, things have changed in my own career span, which is fascinating. But at that point, we were taking individual crypts, we were laser capturing out individual crypts from paraffin samples, and then using nested PCR, so multiple layers of PCR to be able to amplify up the very tiny amounts of DNA that we got out of each of those crypts. And then we were able to do Sanger sequencing of the mutated regions in APC, P53, P16, other genes of interest. And we were able to identify within individual crypts across different lesions what mutations were present in each space. And what you can then do is kind of sum up what mutations are present perhaps in the normal tissue, what mutations are present in the low-grade dysplasia tissue, the pre-cancer tissue, and what mutations are present in the cancer. And by doing that, you can reconstruct the phylogenetic tree. So if for example, you have a PC mutation across everything, but you only have a P53 mutation in the cancer, you can say that at the point of cancer initiation, P53 was a key mutation at that point. So you had that ability to reconstruct phylogenetic trees, which we did many years ago, actually back in 2010, with this, quite unsophisticated technique at the time. As time has gone on, people have used next generation sequencing to be able to look at the entire genome across tiny amounts of DNA. And therefore, the phylogenetic trees that we've been able to reconstruct subsequently have got a lot more sophisticated. And clonal ordering really sort of refers to that ability to be able to generate a phylogenetic tree to say, this mutation happened at this point and led to this pathological outcome.
Mm-hmm. And this sort of trajectory analysis is quite often looked at nowadays with spatial transcriptomics. Do you use that a lot in your work?
Yeah, we're really excited by spatial transcriptomics. In fact, spatial biology is a huge focus of the work we're doing now. So spatial biology refers to understanding the omic expression in the context of the tissue. So rather than with single cell analysis, where you have to disaggregate the tissue, change the structure of how the cells are arranged, but it will enable you to identify cell populations. The beauty of spatial transcriptomics and spatial biology technology per se, is that they allow you to understand or omic expression or mutation analysis within the context of the tissue. So all of those things that we were interested in doing back in the day with laser capturing out individual crypts, you can now see across every single crypt on an image. And the complexity of these tools is increasing. The technology is rapidly expanding. Very exciting opportunity to be able to understand disease biology, within the context of the tissue. And I think it's potentially transformative. But there is a lot of work to do to understand how to analyze and interpret these very large data sets.
Yeah, it has huge power as a tool. Has it been used to measure the spectrum between the crypt-based columnar stem cells and then versus the dedifferentiated regenerative stem cell populations within the lesion?
Yeah, we're very interested in that. So that refers to the idea that there are probably multiple populations of stem cells in tumors. So we have a very long-standing evidence that there is a population of cells called the crypt-based columnar cells, which have bona fide stem cell potential, both in homeostasis and in cancer. But actually, fascinatingly, there are other phenotypes of stem cell that are not shared by the crypt-based columnar cells, and we think that these stem cells can probably interchange. There is the adaptive interchangeability between stem cell phenotypes, and that again comes back to this concept of the tumor as an ecosystem, because it's not just the epithelial mutation that determines what your stem cell phenotype is. It is also the external signaling that cell is subject to. So for example, if the stem cell was surrounded by stromal cells taking in a lot of TGF beta signaling, taking a lot of interferon gamma coming from the immune cells, it is much more likely that stem cell will become a regenerative, somewhat quiescent stem cell, and it can adapt in the niche that it finds itself in. It adapts to the niche that it finds itself in, driven by that niche. And therefore, we have to think, when we're trying to treat cancer, that we're not just dealing with one type of population of cells, we're dealing with a multiple population of cells, a moving target to some extent. And we need to identify the adaptive signaling processes that mediate that phenotypic shift and try and target them too. So as we learn and understand more about cellular phenotypes using the tools like spatial biology, omics, single cell, then we can start to understand how cells change from one cell type to another and what the signaling pathways that mediate that adaptive shift are. And that's really where I think we might need to focus our drugs.
Absolutely. And on the topic of therapeutic side of research, you mentioned earlier the BMP antagonists. So we'll talk a little bit about that now. So you've recently been involved in researching this antagonist called Gremlin-1, which promotes ectopic stem cell niche generation via alteration of this epithelial mesenchymal signaling. But now there's been these novel anti-Grem1 antibodies, which have been developed that reverse this niche generation and reduce polyposis in hereditary mixed polyposis syndrome, or HMPS, in mouse models, at least for now, thereby presenting a preventative strategy to cancer risk. But do you think this has potential as an anti-cancer therapy once these cancerous lesions have actually been established and developed?
Yeah, that's a really nice summary of a sort of complex scenario. So absolutely right. So what we see in a very rare polyposis syndrome called hereditary mixed polyposis is that they overexpress an antagonist of BMP. So back to our analogy of the balance between Wnt and BMP, if you have too much antagonist, you have reduced BMP, which means you have sort of an excess of Wnt, which then drives the formation of what we call ectopic stem cell niches. So stem cells appearing in places that they shouldn't, rather than just being restricted to the base of the crypt. So that sort of ectopic stem cell behavior mediates the the attraction of or the acquisition of mutation, and off those lesions go. So what we found when we treated with an anti-gremlin antibody is that we can reverse the ectopic stem cell phenotype, and fascinatingly, in some recent work that we've done, it suggests that that is actually by reverting the formation of a mesenchymal niche. So again, rather than reverting the epithelium, we're reverting the microenvironment that is pro-stem. So again, thinking about the intestine as an ecosystem, and if you revert part of the ecosystem, you can revert the other half of it, and therefore that that has real, what we think, very exciting preventative opportunities for those rare numbers of patients with this nasty polyposis syndrome. Now, because we talked earlier about the idea that there are multiple types of stem cell, we think that gremlin is a protein that promotes the stem cell phenotype. So there is a clinical trial that is going on to look at an anti-gremlin 1 antibody in tumors that are making a lot of gremlin, in the hope that it might be one of those adaptive pathways that we just discussed, the idea that is an adaptive pathway that promotes stem cell behavior. And if we can reduce the pathway that's promoting the stem cell behavior, we might be able to fix our stem cell phenotype, which will allow us to then attack it with drugs. The problem with drugs is that tumors become resistant to them because they can adapt and they can shift their phenotype in response to the drug. And we hope that gremlin might be one of those proteins that's involved in mediating that adaptive shift. So we're very excited about the trial that's going on at the minute.
So targeting this GREM1 might have, use once the cancer's been developed, not only as a preventative strategy.
I think that's the hope. Yeah, I mean, that's the trial setting that is underway at the present time. So there's a phase 1 trial looking at the safety of this anti-gremlin 1 antibody, but the idea of how that is being deployed is the idea that it will be used in established cancers to try and reduce the adaptive change in stem cell phenotype by reducing one of the stromal supporters of stem cell behavior.
That's quite amazing, because GREM1's also been suggested to regulate FGF receptor signaling in prostate cancer, and also this epithelial mesenchymal transition that happens in pancreatic cancer. So it would have really quite a large impact on the field if this works.
Yeah, I mean, the antibody is super. It's a really effective antibody, and you're absolutely right. There is this sort of potential for a pleiotropic effect of this very unusual protein. It's only 200 amino acids big. It's not a big protein, but it does seem to have, or there is certainly literature evidence to suggest that it has this very pleiotropic effect in cancer. So we're hopeful that it will be a positive trial and will carry forward.
Amazing. And on a more general level, because BMP signaling can oppose the Wnt pathway, as we talked about, in this normal homeostatic state, and promote differentiation, what's your opinion on modifying the BMP pathway as a therapeutic strategy moving forward? Do you think Sorry.
Yeah, no, it's so. Does it have potential? Yes, it's a very, it is a very exciting space. So modifying homeostatic pathways, comes with challenges because they're homeostatic pathways. So they're important in the, the maintenance of your normal gut, and in fact, lots of other normal organs as well. So modifying pathways that will only attack the cancer and not affect your homeostatic tissue is not always easy. But yes, we're doing lots of work with colleagues around the idea that the BMP pathway is an important pathway that can be disrupted to initiate cancer and also as cancer progresses. And again, thinking about those pleiotropic effects, thinking about the effect both on the epithelium but also on the immune cells and on the stromal cells as well. So I think that kind of that multifunctional aspect of these signaling pathways is really exciting and does provide therapeutic opportunities.
Incredible. Maybe it has potential to enhance the effect of conventional chemotherapy rather than as a standalone therapy as well.
I think that's exactly where it's likely to be used, Martha. So I think, yeah, we're approaching the era, I think, where we're moving towards understanding more about the biology of people's tumors, and therefore then attacking that with multiple drugs that are focused at the individual tumor biology. So at the minute we give anti-proliferative chemotherapies that really just stop proliferating cells proliferating, and that doesn't really tackle the underlying biology that might be going on in that tumor. It doesn't prevent those adaptive shifts. It doesn't help us think about how how to tackle both the abnormal epithelium, but also the abnormal environment within which the epithelium finds itself. So I think we are moving towards a kind of multifunctional drug therapeutic approach, where perhaps we have a drug that attacks the epithelium, a drug that attacks the signaling between the stroma and the epithelium, and of course immune checkpoint, which is immunotherapies, which have revolutionized cancer treatments.
That's really exciting for the future. So we've come to our final question now. So to finish things off nicely, what's something that's really exciting you now in the field of stem cell biology, which you think holds great potential for the future to push things forward?
Yeah. So I think I guess the thing that I've been working on a lot very recently with colleagues here, like David Church, who shares my office, we've been working a lot on vaccines. So we're thinking about vaccines not necessarily in the setting of cancer, but in the setting of pre-cancer. So we're talking really about vaccines that can skew your stem cell outcomes at the very earliest stages of initiation. So if you think of an individual stem cell that acquires a mutation, changes its microenvironment, as it's doing so, changes the way it interacts with the immune system, changes the way it acts with the mesenchymal cells. What we would like to be able to do is use a vaccine to prime immune cells to target those initiated stem cells at the very earliest stages of cancer initiation, for use in people with things like Lynch syndrome, FAP, familial adenomatous polyposis, these cancer predisposition syndromes in the gut that cause you to have a very large number of polyps and therefore increase your cancer risk. If we could target those polyps at the very earliest stages with using, utilizing the power of your own immune system, the hope is that by vaccinating people, we can significantly reduce their pre-cancer burden and prevent them from developing cancer in the first place. So we're very interested and excited by pre-cancer vaccines here in Oxford.
That's quite incredible. Thank you so much. This has been amazing. I've really enjoyed reading about your research and talking with you today. So good luck with your future. We can stay on and have a bit of a chat, but for our listeners, thank you so much for listening, and thank you again for today.
Thank you, Martha. It's really nice to talk to you.
Thank you.